Welcome to Chautauqua People. I'm John V. Our guest today is Dr. Courtney Wigdahl-Perry, an associate professor of biology at Fredonia State. She's a native of the state of Wisconsin, where she did her bachelor's and master's degree. She received her PhD from the University of Maine. According to her university webpage, her research interests include lake ecology, paleoimmunology, diatoms, zooplankton, habitat zonations in lakes, saline lakes, harmful algae blooms, water quality, water chemistry, and phytoplankton. She makes her home in Fredonia with her husband, son, and a dog. And she's been slotted as the keynote speaker during several recent Lakes conferences at Chautauqua Institution, and is regarded as one of the most knowledgeable people, if not the most knowledgeable, about the quality of the water in Lake Chautauqua. What experience led you to such research interests?
I grew up around the water. I grew up near Lake Michigan, just a few blocks away, and so I've always been outside and would go fishing with my dad, and we would go down to the beach in the summertime when we were kids. And so I always have enjoyed doing activities outside and near the waterfront. And when I was in college doing my bachelor's, I had an opportunity to take a limnology class, which is a lake ecology study of inland waters. And I was invited by that professor to do a summer research fellowship that was supported by an endowment at University of Wisconsin-La Crosse. And I went out to Montana and Wyoming for a few weeks over the summer, hiked up mountains and sampled lakes, and I was hooked. So it was a really great experience early on in my college career that showed me that that was an opportunity I would really enjoy.
Great. How did you make your way to Fredonia?
I came here for the job at the college. So when I completed my PhD and my postdoc in Maine, I was on the market looking for a job that combined scholarship and research with teaching. I really enjoy the teaching component of the work that I do. So Fredonia offered that opportunity in the biology department.
And the doctorate was in what field?
Ecology and environmental sciences.
And the postdoc done where?
At the University of Maine.
Okay.
And I did a research scientist position as well with Acadia National Park's nonprofit called Friends of Acadia.
Mm-hmm. I wonder if you could tell us a little bit about the lake. I think we have some very great PowerPoints that would give us illustrations.
Okay. Yeah, so Chautauqua Lake is about 13,000 acres.
Let's take a look here.
Okay, great. I have a cheat sheet here.
Yeah.
So Chautauqua Lake is a large, shallow system. It was formed at the end of the last ice age, different parts of it at different times, but generally around 14,000 years ago. It has about 100,000 acres of watershed, and the surface area is about 13,000 acres of the lake itself. And that's split pretty evenly between the upper North Basin and the South Basin. Mm-hmm. The North Basin is a bit deeper. Overall, it's pretty shallow for its surface area. A lot of other lakes that you might see this size are a couple hundred feet deep. But for Chautauqua Lake, the maximum depth is about 76 feet in the North Basin, and then the South Basin is maximum depth of about 21 feet. Mm-hmm. Estimates range a bit depending on where exactly you are and how sediment moves, but it's generally fairly shallow for the size of the lake.
How long does it take for the water to pass through, circulate?
The flushing rate is, depending on which portion of the lake you're talking about, I believe it's somewhere between 6 months to a year is estimated based on how much water comes in and how much leaves.
Mm-hmm. What have you learned about temperatures of the lake?
Temperature in the lake is such an interesting component because it really sets the physical stage for so many other things that are happening. And one thing that happens in many lakes, not just Chautauqua, but in the summertime there's a warm surface layer that forms over a cooler, deeper layer. And we call this process stratification because these two layers don't mix in the summertime.
Really?
Yeah, they stay separate. And what that does is it sets up these completely different environments in that warm surface layer versus the colder, deeper layer. Mm-hmm. Different in terms of not just their temperature, but oxygenation, chemistry, nutrients, pH, how salty the water is, and then what lives there, right, is going to be different because of all of those different characteristics. So what's interesting about Chautauqua is that we do see that stratification process in the North Basin in the summertime that's pretty stable. So once those layers set up sometime in June, usually, year to year it's different, but usually in June it sets up. That will stay until sometime in September often. And then that'll turn over. The lake will cool down enough in the surface layer that the layers can mix together. And they do what's called turnover for the fall and mix everything back up. And then if we have a year where we have good ice on the lake, it'll inverse stratify. So it'll actually be the warmest at the bottom of the lake during the winter. We mix in the spring and it sets it up again. So that's the North Basin. The South Basin is too shallow to stably stratify all summer long.
Yeah.
It will sometimes set up for a little while if it's a calm stretch, but then it'll usually mix back up again within a day.
Mm-hmm. Now, does this make for different types of fish and plants in the lake?
Yeah, because there's all different sorts of biota. Anything that's living in the lake is going to respond to that sort of different physical conditions, right? And so we'll find different sorts of fish that really like that sort of colder refuge. They like to hang out right along that boundary, like which we call the thermocline, that boundary between the two layers. So often, like musky fishermen will ask me, where's the thermocline sitting? Because they'll want to know where the fish are riding along that line where we have that boundary between the warm water and the cool water.
So the muskies then make for the boundary.
Sometimes I'm not a musky fisherman, so I'm not an expert there. But oftentimes they'll like to hang out where it's a little bit cooler. But also they're like in the deeper water, there's not very much oxygen, so they can't stay down there very long. Yeah, in Chautauqua. Mm-hmm.
Got it.
And then in the shallower South Basin, you're going to have different sorts of species of fish as well because there's more plant habitat for them to be moving in and out of, and different kinds of conditions in terms of water clarity and mixing patterns and all those things affect which species are going to be most successful.
How does the oxygen get into the lake?
Oxygen gets into the lake from the physical agitation of mixing in and diffusion from the atmosphere into the water, but then it also gets in there through plants and algae performing photosynthesis.
Really?
So taking energy from the sun and changing carbon dioxide into sugars, and that byproduct is oxygen, which is a handy product for us, but it's just sort of an accident as far as photosynthesis goes.
So there's another process beyond the icing during the winter. Mm-hmm. Several processes then
that change the
oxygen level.
Yeah, so we've got different balances, and in the summertime, the reason why we have in a lake like Chautauqua we have low oxygen in the bottom layer is because the balance to photosynthesis is decomposition. So something dies and bacteria start to break it down. That process is oxygen hungry to be more efficient, right? Breaks it down more efficiently if there's oxygen present. But that causes all that oxygen to get sucked up. And since there's not a lot of light down there in those deeper portions of the lake, and there's not enough agitation or diffusion from the atmosphere, that boundary stays separate. And so once the oxygen is depleted from the bottom portion of the lake, it stays that way until it gets mixed up again.
Wow, what a story. Mm-hmm. Now, does the oxygen level vary from year to year?
It can vary from day to day.
Yeah.
It's just really also interesting. The surface levels will respond based on the algae growth, typically like in the open water. Mm-hmm. So if you have a big bloom, you'll have what we call oversaturation. You'll have this really high oxygen percentage. Mm-hmm. And then if there's not a big algal bloom, it'll usually be a little bit lower. And then you'll see, once the layer's set up, you'll see oxygen present in the bottom layer for a little while, and then it usually gets depleted fairly quickly.
Really?
Mm-hmm. It happens fast.
Can we take a look at the second view graph on the lake?
Yeah.
Tell us about what we need to know here.
Yeah, so again, we have sort of a long, skinny system, which is not unusual in glacially formed lakes of this size. It's just kind of how the glacial lobe retreats and leaves these scars. There's lots of ways that lakes can form, and this is one of them. It's 13,000 acres, like I said before, about 20 miles long. It has about 42 miles of shoreline. It has a large, complex watershed, and you can see here and in the slide before that there's different streams that are coming in and feeding into the basins. And so we're sourcing a really, not an overly large watershed, but we're sourcing from lots of different parts of the landscape around the lake as that water's coming in. And what's interesting about the system is that it's a headwater system to the Mississippi River drainage. And so you could, in theory, start here and take a canoe or a kayak all the way down through the Allegheny, down to the Ohio, and out to Mississippi. So even though we're close to the Great Lakes drainage system, we don't drain out that way. We drain out completely different direction. And so it's a headwater system, meaning that there's no standing water above it. So it's the first sort of stopping point, first pause point of water flowing through this drainage. It's what we would classify as eutrophic, which means productive. And what that means is it has more photosynthesis, more plant and algae growth that happens in the system. And in part that's due to its shallow nature because it tends to be warmer, and so that increases how fast things can grow. And also we have a fair bit of nutrients. We have a really rich organic landscape over the long history of the lake, and more recently based on what humans are doing on the landscape as well.
Now, how many years have you been doing research on the lake?
I've been here since 2014 at Fredonia, and so I started driving around and looking at lakes pretty much as soon as I arrived.
Wow.
So I've been actively doing stuff probably since summer 2015.
So 11 years of research.
Mm-hmm.
Have you seen changes in the lake from year to year?
Every year is really different.
It is.
I think every year is different. I think that's one of the things that's, as a scientist, really fascinating about this lake is that every year it feels like there's something new that we have to learn. It was a very different spring, or we had a really big drought, or we had a strange growth pattern. We had a bloom here, and then the next year we had nothing, you know? And so it does tend to vary quite a bit from year to year, and not necessarily in a steady direction, which makes it tricky.
So it's difficult to make predictions then?
Very much so. Mm-hmm.
Somebody would always say, go develop a model, but what's the model worth if it has no predictive value?
Well, it's models help us understand the system, right? They help us understand why something happens. And then once you have a working model, you can use it to forecast. We have to have enough data and enough of the right kinds of data to develop these sorts of models. And they have to be designed with really specific predictions, right? Or specific goals, I should say. So we want to make sure that we have, if we have a good working model, which other research groups are working on right now, for nutrient budgets, for biological models, those sorts of things. But it's a big undertaking, and everybody wants that forecast, right? But we have to understand this really complex system first before we can make predictions going forward.
And where does most of where does most of your research funding come from?
I have a pretty tight budget, so we've gotten funds from a variety of sources. We've had support from federal grants, state grants, local donors who have been very generous. And a lot of my work, because we're primarily a teaching institution, is meant to be really streamlined and work on a pretty tight budget. So we have very generous endowments that support our student researchers, and a lot of the stuff we get done over the years has been through that. So we've leveraged a lot of different fundings from different pockets. When I first got here, I did a SUNY startup grant, a 4E grant, with some colleagues at other SUNY institutions, and that was a lot of what sort of kicked off some of the sensor data we were working on early on. So we've had fantastic support from a number of different arenas over the years.
Grant writing is hard work, isn't it?
It is, yeah. But it's fun to tell the story and frame the question and think about what do people get invested in, what do they care about, how can I get my science to line up with what people want to know so that I'm still doing really cool science, but also something that's going to help people, something that's going to change what somebody knows about their system.
Mm-hmm. Now, tell me a little bit more about your students and what their data collection is.
So I am pretty heavy teaching load, so we do a lot of teaching at SUNY Fredonia, but we really value these student experiences, the experiential learning opportunities that we have. It's something we're very good at providing to our students. And it makes a big difference to have students doing authentic research. You know, there are labs that go along with our courses, and labs are there to teach you these skills, but they don't necessarily show you what real science is like, because most of the time it doesn't go the way you expect. And so real authentic science happens when we're hitting walls and we're making mistakes, or we're learning something new that we weren't expecting. We didn't support our hypothesis for that experiment. Why not? Is that real, or is that something that we didn't do correctly? Like, what is the question? And so my students have really been, from the first projects we did, the very first project I did down there was a master's project with a student, where we were adding nutrients to these lake samples and watching to see what caused the algae to grow. And it was fascinating, and that was, thinking about how much the lake changes even over the course of a season was really eye-opening for me, just learning about the lake for the first time. And so my students are really active in the field, collecting samples, but also in the laboratory.
Look at this.
I'm so lucky to work with so many great students. And so we're out for I teach a class called aquatic biology, aquatic ecology. And so we take students out and do field sampling, and we have...
Can you talk us around
sure.
the corner here?
Yes. So we do, again, lots of different activities in the field. And so I have students in the upper two pictures pulling sediment cores. So that's the paleolimnology reference.
So they have to go down to the deepest spot then?
Yeah, so we'll take this tube that you can see there. It's just a clear plastic polycarbonate tube. And we have different methods, but we get it down to the sediment, and then we pull up this tube of sediment. And essentially what lives in the lake falls down to the bottom and becomes preserved there over time. And so by looking back through the sediments, we're looking at a naturally occurring archive, a record of what has been there before. And so my students go out and help me collect and for their questions that they're asking, and then we process it in the field and bring it back to the lab.
Now let's question how many years back?
Oh, I love that question.
Sediment.
I don't have an answer. We have done some pilot work over the years to see what the sediments are like and make sure there's actual preservation of the things we're looking for, and it looks very promising. But Chautauqua Lake is a really productive system, so a lot of material probably gets deposited and preserved every year, which means that we have to go get a lot of mud to make sure that we could get a good date on it. There's a lot of technical components, but basically we have to make sure, if we're going to do this dating approach, we have to get back at least 200 years.
200 years.
And that could be 3 to 6 feet of mud.
Right.
And so it's a big push to go back that far, and we can absolutely do it.
So you put them, I don't mean to interrupt, but you pull a core then?
Yeah.
A straight core?
Yeah, it's kind of like an old push pop.
Whoa.
You push it up from the bottom, and sometimes you keep the whole core intact, but what we do for my lab is we scrape it off in little sections. And so we have these little mud cookies that come off the top, and then we work with each of those individual samples. So it's a big investment to get the time into that, to get all the information out. And right now where we're sitting is we've done pilot work over the last few years. We've had some interesting questions we had to resolve first with this type of algae called diatoms, which have a glass cell wall and this really beautiful ornamentation and different patterns. But these algae are found all over the world in all these different environments. And what's helpful about that is we know where they like to live now, which means that when we find them in our cores in the past, we have a sense of what the environment was probably like before. And so they're an indicator for us. But we had some time we had to spend figuring out all these different species that look very, very similar before we could feel confident and okay, I think we're ready to move forward. So I think I had some students who did some really nice work on that project, several different students who've done the cores over the years. There's another core on that, that set too.
So that's a core in the middle?
Yep, that's another core in the middle.
Okay.
And that was the second one we took from Chautauqua. So that was, I think that one was from the North Basin. And so we were, we've done these pilot cores. They're not always easy to get. And so now we've had a couple years under our belts with multiple students working on that project to tell us more about the history. So at this point we feel good, and it's just a matter of putting a story together to get some more funding to go after a bigger project that would involve more of that dating. So that amount of mud, like this amount of mud in some lakes would be thousands of years, and at Chautauqua that might be 10 years.
Really?
It varies a lot. So I don't have an answer for you for getting back to your original question. I don't have an answer for the time, but I'm guessing, a couple hundred years there.
So am I fair in saying that the Chautauqua Lake is accreting at the bottom at a higher rate than similar lakes?
Yeah, because if you think about if what deposits on the bottom is all the stuff that's lived there,
Yeah.
if you have a lake that has a lot of stuff that's active every year and it sinks down to the bottom, just more material gets piled up every year. Other lakes that don't have as much activity going on, they don't drop as much stuff. And so some of like the high elevation mountain lakes that I've worked on, they don't put much down every year. It's really, really minimal. They're very, very clear.
Really?
Yeah.
And so there's less,
There's just
because
less
there's less
stuff.
vegetation.
Yeah.
That's significant.
Yeah.
Now, what do you say to somebody who they complain they want to go swimming in Lake Chautauqua and it's not as clean as their swimming pool?
Well, it's just not a swimming pool. It's a living, dynamic lake. It's a beautiful system. It's complicated. I often get asked, when are you getting rid of the algae? When are you going to make the algae go away? And it's really about seeing the whole story. So if you enjoy having world-class muskie and bass fisheries like we do at Chautauqua, you need a lot of energy coming into the system. You need a lot of that base of the food web. And without that, the big fish can't get really, really big. And if you want to have those fisheries, you need to have algae and plants growing. That's just how you get there. So it's important to just remember that it's a lake. It's not a swimming pool. It's not. It's going to have always stuff living in it. I think we're really used to the sense of control when we have swimming pools. Like if you have if your swimming pool turns green, you just dump your chemicals in it and it resets and it's fine the next day. But that's not how a real dynamic system works. And something is always going to grow in it, right? If you leave a bucket of water on your driveway or in your yard, right, something is going to colonize it and grow in it pretty quickly. So we'll never have a lake that doesn't have anything growing in it, and we don't want that. We want something that's a functional, resilient system. But it is frustrating when people want to use the lake for certain purposes and then they're not able to do so because of concerns about health or just the quality or aesthetics. And I think that's the tricky part about management, because everybody has different goals or objectives or values that they place on the lake and what they want it to look like. And that comes from all of our different personal experiences that we each have. And nobody's perspective is going to be identical. And it's hard. I appreciate the frustration. And if you spend money on a home on the lake and it doesn't smell very good when you open your windows in the summertime, or you have a business that's affected by whether there's a bloom in the lake or whether there's plants growing, that's all very real and very impactful to people's lives. It's just never going to be a swimming pool. There's always going to be stuff living in it.
Now, with your 10 years or a decade of field research on the lake and pulling up these mud samples that are many, many years old, can you see how the lake has changed over time?
It's interesting when we look at, there have been changes, there have been shifts in the species that we found. What that means, hard to say. Probably the records that we're looking at are probably 50 to 100 years old, so we're not getting a full picture before human activity was really impacting and shaping the lake. So ideally with a study like that, we would be able to go back well before we had modern architecture and roadways and septic systems and things like that, and agriculture in the area. There have definitely been some trends in the last 50 to 100 years that we've seen, but I can't tell you exactly what that means without more context for where we started. In terms of our other data sets, we have, we're very fortunate to be part of the CSLAP program, the Citizen Statewide Lake Assessment Program. CSLAP, Citizen Statewide Lake Assessment Program.
Okay. That sounds like something older.
I know. It's a fantastic program. It's New York State's volunteer monitoring program. So lots of states have different programs kind of like this. But CSLAP started in 1986 or 1987, and Chautauqua was one of the first lakes that was in that. So we actually have some really wonderful records collected by volunteers and supported by the state. People going out every other week for the last 30 some years during the summertime, just volunteering to go out. And so we have these really great records for a lot of data going back. Every year, CSLAP does an analysis of what the trend is, what the pattern is in these key variables. And there's good reasons why it might not be the whole story, but based on those data, which is our best longitudinal long-term record, there's very little consistent change in algae biomass, in nutrients for phosphorus, a little bit of an increase, and for water clarity. Sometimes it goes up, sometimes it goes down, and we look at the last 10 years, but there's interesting year-to-year changes. The variability is high, but not really strong directional trends. So for example, last year when I had done a talk with the lake conference at the institution, I had pulled up the CSLAP report and there was no trend, no trend, no trend. And then this year when I pulled up the report, there was a trend. So like 1 year could actually pull up and have a trend, and then the next year it'll go away. So that just kind of tells you it's a variable data set.
Those are statistical models that would determine a trend or not.
Yeah. So what we are doing is accruing that information over time, and we also have more people working on the lake than ever before, really, in terms of different research activities and projects that are going on. And so we're asking different kinds of questions. We're extending the season that we can sample from, so we have more people going out earlier and later than we ever have before, which I think is helping us answer some of that seasonality, like how the lake changes over time, and why is it this way 1 year and not the next. And so and more spatial sampling too is the other thing I really learned to appreciate about Chautauqua Lake. Is as lake people, lake scientists, we tend to take like one sample in the middle of the lake and say, that's it. That's our representative sample, because it's so much work associated with just processing that for one location.
Mm-hmm.
And I don't know that people see it. They don't see the back end of it,
Yeah.
right? But I tell my students 1 day in the field generates, at least from one location, generates probably 3 weeks to 2 months worth of work for us in the lab.
Can you tell me a little more
Sure.
about the student programs at Fredonia?
Oh, yes. So we have students that are working we're in the biology department. I'm associated with the biology department and the environmental science program in the geo and environmental science department. And so I work with all kinds of students interested in ecology and the environment, and other students that are doing different kinds of things. So I have a range of majors that have joined my lab over the years, which is really fun. I've had education students, so people are going on to be teachers. I've had students who are biochemistry majors, biology, environmental science, like just a range of students and what they want to do. And so I get to teach them in classes during the semester, and then also work with them in the lab. So I mentioned the experiments and the paleo work. I've had students working with sensors, temperature sensors mostly, and oxygen sensors. They'll collaborate with other people that I work with to do different kinds of projects as well. So sometimes it's experiments and work in the laboratory and time on the microscope, looking at algae samples. I've had students do experiments with zooplankton sensitivity to road salt was another cool one we did. And other students who've done some really great like modeling work with the temperature data and some sensors we've put out. So I think what's really exciting for me about that is in my role, because I'm not just research-based, I'm not just scholarship, I'm really focused on teaching, is that I can let the students ask what they want to ask. And I don't always know the answer. In fact, I usually don't. And sometimes I don't even know, I've not done that protocol before. But it's a really great experience for us to do that together and to show them, like, you don't have to have the answer. You need to know how to find the answer and how to work the problem and figure out how to get information that you need to apply it to the situation. But I have branched out in some really cool areas that I probably wouldn't have picked for myself. I probably would not have done as much work as we've done with the stratification and the temperature layering if I didn't have a really dedicated student who was really curious about that. Actually, a couple. I've had a couple, and they really pushed it, and they came up with their own original ideas and said, I really want to tackle this. I said, okay, great. I don't know what you're going to find, but let's do it.
Now, typically how many students a year are involved in such work?
This past year I had 7 students working with me in the lab under various projects. Some years it's 2 or 3, sometimes it's 7. So it ranges depending on the year and the different kinds of projects that the students have going. So I have 3 I had a big class that I had 6 of my students last year graduated. So I have 1 that's continuing working with me this summer, and then I have 2 more students that'll be joining the lab this fall.
What becomes of most of your graduates? What do they do?
They are very successful. They've gone on to PhD programs at large research institutions. I've got some that are working at the county health department, other students that have worked for consulting companies. I had my first graduate student actually changed gears and is working in the grape industry now, which is great. And I have another one, and she's doing fantastic. I have another one who went on to do his PhD in food science at Cornell. And so they're ranging all over the place in terms of I have some that work for the DEC and some that work for the DOT as well as environmental specialists.
So I'm left with the impression there are jobs for your graduates.
Absolutely, yeah. We always need clean water to drink. We need to have the ability to build safe infrastructure over and through these environments and protect those resources and follow those regulations. There's a lot of opportunity, and I think sometimes environmental issues ebb and flow, but we live in this space and we need to take care of it, and that becomes really evident in certain cases, and so there's a lot of opportunity. And I think one of the great things that we do is we train our students to be really broad-based. They've got a fantastic foundation, and so all these students took similar classes, right? They all come from the same home base, and yet they're going out into all these really wide-ranging, some nonprofits, state agencies, federal agencies. They're going all over the place into academia. But they're starting from that foundation. So we train them well to be prepared, but we also really listen to what they're excited about and try to find research opportunities, internships, things that line up with their interests so that they can tailor not just their elective classes, but their hands-on experiences to get them where they want to go.
That's great. I got one question I'd love to ask.
Yeah.
It's sort of a conclusion. We're over time. The staff always indulges me. This is so much fun. I can't worry about the time. Let's say you're on the grounds of Chautauqua Institution. You gave a presentation a couple of weeks ago. Some young Chautauquan sits down with you and says, could we talk? And that young Chautauquan says, I would love to have a career doing marine biology, the sort of things you've done. What advice would you give that Chautauquan?
I would say, go for it. Just find what you're excited about, find people that support you. Don't let anybody stop you from going after it. I knocked on a lot of doors, I asked a lot of questions. Most people were really happy to help me along the way. Some people were less helpful. But I found that talking to people and building those relationships really got me through and really helped me become effective at this. And so I think just going for it and not being afraid of failure. Don't be afraid to fall on your face, because science is all about failing.
Yeah.
And a lot of that's knocking on doors and getting to know people. We as scientists don't always talk well to, we're not always super social, but that's a big part of it. And putting yourself out there and going for it is really, that perseverance part of it, I think, is what we always joke about in academics. Is like, being successful in science isn't about being the smartest person in the room. It's about, are you willing to stick with it? Are you willing to keep fighting to solve the puzzle long past most of the time when anybody else would have quit? And eventually you'll find the answer. You just have to be willing to stick with it and persevere. So you just got to go for it.
That's great. Thank you so very much.
Yeah, of course.
And please do come back.
I would love to.
In a few years, and let's talk about the research since then. These are some fascinating issues.
Yeah, it's a great place to work. It's really interesting, great people who are really invested in making the lake as great as it can be going forward. So it's a wonderful place to work.
Thank you so much.
Thanks.